Andrew Holland, Director of Oncology Research. My favorite piece of equipment is a microscope. I was trained as a cell biologist, and a microscope allows us to peer into the inner workings of a cell. And one of the areas that I focused a lot of my career on was trying to understand how cells divide, how one cell becomes two. And what we can do with a microscope is watch that in real time, and we can visualize individual proteins and structures inside of the cells and watch those proteins transform and ultimately perform the job of splitting the cell. So my favorite model organism is the mouse, and the reason for that is that the mouse has physiology that is fairly similar to that of ourselves, so we can learn a lot about human biology from a mouse. And also there's just an amazing toolbox available for interrogating biology in mice. Most notably, you can manipulate the genome of mice and perform precision experiments to learn about how that genetic information encodes phenotypic information in that animal. Yeah, I don't think I had a particular role model that I could point to. But one thing I think that did define my early childhood was I was curious about the world, how things worked. I like to take things apart, put them back together again. And I think that drew me towards science as a child, because science ultimately gave me questions and answers to what was happening around me. And it was a lens through which I could understand and rationalize the world. So I really enjoyed the fact that what I learned in a classroom seemed to have direct implications to things that I could see and observe around me. And so I think it was kind of a natural fit for me to become a scientist. Yeah, I think as a young child, up until 11, I went to a local primary school, and I certainly wasn't thriving there. My parents took note of that and decided that they wanted to send me to another school that was quite away from where I lived. And it was a private school, so they saved quite hard to be able to provide the funds for my tuition. And I would travel to this school that was maybe an hour or so ride on 2 buses to get there every day. And I think that was a big transformation in my life and, my journey, because I experienced a very different type of teacher there, people who were very dedicated. There was a lot more opportunities for me. And I really began to grow as a person and ultimately began to love science. And by the time I was 18, I was thinking about going to college, and I was trying to think about what I wanted to study. And in the end, I chose to study a degree that was called a natural science tripos, because I could study all 3 disciplines of science, chemistry, physics, and biology, because I didn't really have any specific idea of what I wanted to do with it. I knew that I just wanted to continue my education in science. Well, no, I'm definitely firmly in the biology camp now, but I studied a lot of chemistry at college. I wasn't a very sophisticated kind of student. I hadn't had any experience with research. I didn't know a lot about what I was going to do with my scientific education. And at the end of my journey through college, my advisor sort of told me I should do a lab placement for 3 or 4 months as a program before I left. And I sort of fought and argued that I didn't want to do this and that I was better just studying in the library and writing a thesis. But they eventually convinced me that I should do that. And I spent some time in a research lab. I was at the University of Cambridge, and that really changed my view of things. Once I started to spend time in the lab, it was really transformational for me, because it was the first time in my life where I'd had a job where I wasn't looking at my watch wondering when the day was going to end. All of a sudden, things were beginning to click, and it was like, oh, I could see I enjoyed this. I liked the freedom. I liked the curiosity. I liked the fact that you could set your own agenda and try and answer important questions. So right now my field is in trying to develop medicines for cancer. And I think, what we've become very good at as drug discovery individuals, and it is is turning off proteins that are aberrantly activated in cancer. You can think of many of the programs that drive cancer as like signaling cascades, and these proteins get altered aberrantly in a way that pushes the accelerator on and creates a constant signal to these cells so that they behave abnormally. But equally importantly, proteins are often lost in cancer. They can be deleted at the level of the DNA, or they can be mutated in a way where they're no longer functional. And we have limited tools to be able to attack those type of problems, because it's much more challenging to try to restore the function of something that's disappeared than to turn off something that's aberrantly switched on. And so I think that's one of the greatest challenges that we have in drug discovery, is how we can develop creative approaches to try and restore the function of proteins that are disabled in cancer, or more likely take advantage of the fact that when those proteins are lost, the cells behave in different ways that make them dependent on new features of their biology. And we can try to take advantage of turning off those features. And that's something we call synthetic lethality, where the dependency is encoded by the loss of function of something that's critical. Yeah, I think I have a couple of answers to that. I would say that the critical, most profound discovery in science is the theory of natural selection by evolution from Darwin in the 1850s. And that provides a lens through which we view all of biology today. There's a saying that nothing makes sense except in the light of evolution. And, more than 100 years later, Richard Dawkins published a book called The Selfish Gene, which sort of took that theory to the extreme of suggesting that not only are we looking at organisms that are behaving in the way that they do because of the way they're shaped by natural selection, but in fact, the DNA, the instruction manual of life itself, is subject to those very same forces. And the idea was that DNA elements try to propagate themselves and increase their abundance over time. And the sort of most, striking observation from all of that is that we ourselves are just embodiments that are dispensable, that allow DNA to be able to increase in number over time. And, this sort of explains, for example, why a large fraction of our genome is selfish elements that have no known function. They're just there to increase their number and propagate. So, I think that theory of natural selection allows you to see all of biology in a very profound and different light. Yeah, so before I started my current career, I was an academic researcher and ran a research lab. And we did one particular experiment where we took a pharmaceutical, a small molecule, that inhibits the action of a protein. And we'd used this in many contexts before, and it had been very well tolerated. Essentially, the cells didn't really care that this protein was inhibited. And what fascinated me was we took a certain cell line and we treated them with this molecule, and they all died. And all of a sudden, there's some very clear difference here in that these cells really care and the majority don't. And so we sort of try to dig into that and understand what it was that was different in this particular cancer cell line that meant that they were highly dependent on the function of this protein. And ultimately, we uncovered what that was at a genetic level. And we published papers explaining this along with another team from UC San Diego. And that is now moving forward as a practice in a clinical trial where molecules that target this very protein are being trialed in patients that carry these specific alterations that were present in those cancer cells. So that was a very satisfying journey where, we were able to take a curiosity in the lab and move it all the way forward. And then other people have picked that up, and ultimately, maybe one day that'll translate into something that could help people. I think the main thing that I did throughout my life was I followed my interests and passion. And my parents were very good in that they never pushed me in one direction or another. And I took step by step to just follow and do the things I was interested in. And that went from my degree to my PhD to running a lab and now where I am today. And so, following your passion is definitely what I would recommend. And, making sure that it's what you're looking for in life, and that it fits your needs and that you're not trying to satisfy somebody else. I think for me, the biggest set of challenges were when I started my own research team about 10 years ago now, because I went from working day to day in a lab as a postdoctoral fellow to all of a sudden trying to run my own lab. And that came with a whole suite of new challenges and opportunities. So I didn't just have to direct the science as I was doing as a postdoctoral fellow, but I had to mentor people. I had to try to recruit people to the lab. I had to try and raise funds for the lab. I had to essentially advertise. And these were all very new opportunities for me. And, it took some learning, and there was no particular training program that I could take at that time to get up to speed on how to do this. So you rely a lot on the generosity of your colleagues who, will help you through some of the challenges because they've been there before. Yeah, be curious and dig deep. Don't accept things on the face. And what's amazing today and wasn't really available when I was a child is that everything, all the knowledge of the world is at your fingertips on the internet. And so, when you see something and you wonder how that works, take the time to try and understand it, read about it. And, I think that brings a huge amount of satisfaction because that's what, human beings are curious and we want to understand how things work. And I think if you follow that, you'll be able to get very deep in your knowledge of how the world operates. And that's the sort of mantra that I followed.